Hi, everyone. My name is Vardan Ter-Antonyan, and I'm the R&D manager at Mile High Labs in Loveland, Colorado. From isolates to distillates to nanoemulsions and microemulsions, Mile High Labs manufactures and provides highest quality phytocannabinoids in the industry. Before I go into the presentation, I wanted to say a couple of words of gratitude. Thank you so much for inviting me to speak on the phytocannabinoid nanoemulsions and their applications in food and beverage industries. So just a few words about, can you see my second screen?
Only one.
Hello?
Only one.
Only one screen? Just a second. I will stop sharing and then I'll share again. So, introduction to phytocannabinoids. So phytocannabinoids, they occur in cannabis plants, and contrary to the popular belief that anything related to cannabis is associated with getting high, cannabis plant actually contains hundreds of compounds inside. And so but it starts out with only one phytocannabinoid, which is called mother cannabinoid or cannabigerolic acid. The formula is on the screen. It is also called CBGA. So along with CBGA, there are multitudes of enzymes in the cannabis plant. They mostly accumulate in the buds or flowers. So through enzymatic reactions or synthase reactions, CBGA actually degrades or turns into cannabidiolic acid, CBDA, delta-9 tetrahydrocannabinolic acid, or delta-9 THCA, and cannabichrominic acid, or CBCA. So none of these are psychoactive, actually. So then over time, through the process of thermal, and photolytic, and oxidative degradation, these acids could turn into bases and turn into other cannabinoids. There are hundreds of cannabinoids in the plant. So through different enzymatic reactions, there could be different amounts of these acids in the plant. So those enzymes are what determines the strain of the cannabis. So sativa strain contains more delta-9 THCA than CBDA, and indica strain is vice versa, contains more CBDA than delta-9 THCA. Then there are hybrid strains that contain maybe 1-to-1 ratio or other different ratios. The buds, later they're harvested and then they're decarboxylated, so the acid forms turn into the base forms. And so these are some of the well-known cannabinoids: CBD, delta-9 THC, and CBC. Now, the importance of these cannabinoids are that phytocannabinoids are effectively interact with the endocannabinoid cell signaling system in the body. And this endocannabinoid system is actually responsible for very much existential and very important processes in the human body. And so these phytocannabinoids are able to actually generate the same responses, same signals, and so they are extremely important for the human body. But the problem is that these cannabinoids are lipophilic, and their bioavailability is very small in the body. I just wanted to confirm that it went to the third slide, correct?
Yes.
Yes, great. Okay. So the problem is low bioavailability. It's 5 to 10% depending on the diet that a person is having. So if a user has a fatty diet, eats a lot of fats, the body generates a lot of enzymes that break those fats down into smaller constituents, so fats into fatty acids and so on. So the more of those enzymes is in your system, the more efficiently it processes these lipids, the phytocannabinoids. And the less fat you have, the less fat, the less enzymes. So the bioavailability drops to 5% or 4%, something like that. So in order to really understand the problem and know how to solve this problem, we need to look into how the body digests fats, lipids, oils, and just food in general. And, and then look at the experimental data and see how we can come up with the theory on that. So the food digestion starts happening in the mouth. And, as soon as you put the food in the mouth, there's digestive proteins and enzymes plus chewing process. So, some absorption happens in the mouth, and I'm talking about lipid absorption, right? So, however, the food doesn't, or beverages, they don't really spend that much time in the mouth. So not a whole lot of absorption happens. Then everything kind of drops down into the stomach. And in its stomach has very acidic environment, as everyone knows. Digestive gastric enzymes turn, fats into fatty acids, and triglycerides into diglycerides. But in the process usually takes 4 to 6 hours for all that food and stuff to get digested and, pushed forward into the small intestine. Now, absorption in the stomach is also very little, because only acidic compounds absorb go systemic through the stomach epithelial, cell membrane. So, lipids usually just drop into the small intestine. There, what happens is most of the absorption happens there. And so in the small intestine, the diglycerides get broken down into monoglycerides, and fatty acids, as well as the monoglycerides, get emulsified by bile. So bile is composed mostly of bile salts, lecithin, as well as cholesterol. So, but bile salts and lecithin are very powerful emulsifiers or surfactants, surface active agents, and we'll talk about them later. But what happens here is that these, in terms of lipids in the small intestine, there are large lipids and there are smaller lipids. So there's like variety of sizes of lipid droplets that make it from the stomach into the small intestine. And so, all these lipids, they get emulsified by bile. So they get emulsification process, which I'll go into a little bit later, is a process of self-organization of surface active agents, surfactants, around the lipid, the lipid molecules. And so they basically encapsulate the lipid. And these come in variety of sizes in the small intestine. So super small ones, really small ones, called mixed micelles, that are 4 to 8 nanometers in diameter. These are basically spheres, like I'll show you later. Then we have vesicles that are basically sub 100 nanometer, 100 nanometers to 10 nanometers, that range. Then there are liquid crystals that are sub 500, sub 700 nanometer, droplets, or as we call them, micelles. Obviously there are larger ones as well. There's 1 micron, 2 micron, and so on. And experimentally, we know that the smaller the micelle diameter, the faster and better these micelles get absorbed, if they are in the vicinity of the epithelial cell membrane in the small intestine. And so the larger they get, the slower the absorption, and the less of them get absorbed into the system. So it basically equates to, if I were to draw a parallel, it would be basically, if you have a basketball hoop and you drop a ping pong ball through it, that would be the mixed micelles. If you drop a basketball through it, which would go a little slower, it touches the net and the rim and so on. So those would be, vesicles. Liquid crystals would be, a little larger balls. You need a little more work to actually break them down into smaller micelles before they can go in. And even then, they might not, all go in. So some of them will be at certain period of time, some of them will be excreted, pushed forward into the colon and then excreted, because in the colon, there's not much absorption in the colon. So basically, from here, let's talk about how do we solve this problem of low bioavailability. Well, the body solves it. The body solves the problem of lipid absorption by emulsifying it. Well, why don't we emulsify these lipids before we even ingest them? In this case, if they have small diameter and we ingest them in that form, more of them should be able to absorb into the body, so increase the bioavailability. So first, to do that, we need to make oil in water nanoemulsions. So nanotechnology comes to the rescue in this case. So we have our lipids, phytocannabinoids. We can do, let's say, the cannabidiol, CBD. Then you need carrier oil in order to kind of dissolve your lipids in the carrier oil, and then basically incorporate that carrier oil lipid mixture into your system. So the carrier oils could be medium chain triglycerides, long chain triglycerides, could be olive oil, could be any kind of vegetable oil. Any kind of lipid environment would work. Now, the key here is to have larger amount of carrier oil than your lipid. The ideal ratio would be 2 to 1. That's the ideal ratio, in which case you get stable nanoemulsions. Then obviously you need water, because you have oil in water emulsion. But the key here is water has to be, weight-wise, it has to be more water in your system than oil, right? Then more importantly, also surfactants. So surfactants or emulsifiers, they come in a few different varieties, ionic and non-ionic. So ionic ones, the most popular ones, the best or the safest ones to use for foods and beverages would be the zwitterionic surfactant. Most popular one is the lecithin. And then the non-ionic ones are, polysorbate 80, polyglycerol ester, or vitamin E TPGS. That's a tocopherol polyethylene glycol succinate. So these emulsifiers, if you look at their structures, basically what you see is a polar head, which is a polar hydrophilic head, and a non-polar lipophilic tail or tails. In case of lecithin, it's got two lipophilic tails. Then it's got 3 different groups. Now, why is it called zwitterionic? Is because it's got 2 oppositely charged polar groups. So you have choline group, positively charged, then you have a phosphate group that's negatively charged, and you have a neutral glycerol group with 2 tails. And so looking at the polysorbate 80, it's got a giant polar head. It's got only one group. Then you have a 1 tail. The rest of them basically follow the same structure. Now, with lecithin actually does not have an upper limit of use. So the acceptable daily intake for lecithin is infinite. You can eat lecithin as is, and because the body is able to break down lecithin very effectively and use every single group, every single component of this molecule separately, absorb it and use it in the body. So this is extremely safe and natural as well. Polysorbate 80, on the other hand, it's polar. The tail is able to kind of get absorbed and processed by the body, by the cells, but the polar head is not. So these usually, they can lodge themselves into the epithelial cell membrane and just get stuck there. So, their safety upper limit is very high, which is good. So you can use it, but I wouldn't suggest super saturating your emulsion with this polysorbate 80, because then you will exceed that daily intake, acceptable daily intake, and then that could cause toxicity and stuff. So same goes for the polyglycerol esters and vitamin E TPGS. So moving forward, let's talk about how we make nanoemulsions. So we have water phase and we have oil phase. The oil phase is composed of carrier oil, your lipid, in this case CBD isolate, and oil and water emulsifier. So this system gets emulsified by the rotor stator emulsifier. And what you see in this case is basically we have reversed empty micelles. It's self-organized spheres with hydrophilic heads in, kind of tightly together in the center. And then the lipophilic tails are sticking out to interact with the lipid environment in the oil phase. You heat this up usually, to higher than the micelle creation temperature, 45, 55 C. To prepare the water phase, we add water, preservatives, we add pH adjusters, it could be citric acid. You have the oil and water emulsifier, and you then again emulsify it with a rotor stator emulsifier. So in this case, what you get is empty normal micelles. And then the oil phase is then slowly added into the water phase while the batch is being homogenized with a rotor stator homogenizer at a very high shear, high speed, or revolutions per minute. So, what you have as a result is basically micelles. These are spherical structures with the lipid trapped in the core, and on the outside is the hydrophilic heads. And this is basically the three-dimensional picture of how it looks, with the carrier oil and your lipid of interest encapsulated in the middle. However, in this case, this emulsion so far is actually a macro emulsion. It has an emulsion droplet size or diameter of 2 to 3 to 4 micron. So, and that's because of that, it's not stable. So if you leave it for 24 hours unmixed, agitated, it will basically separate. You'll see a distinct layer of separation between oil and water, and we want to prevent that. So in order to do that, we need to decrease the particle size even further, to 500 nanometers or below. The smaller the droplet size, the more stable is your emulsion. So in order to do that, there's lots of ways to do that. There's ultrasonication and other ways, but today I'll briefly discuss the high pressure homogenization. This is a process where the emulsion with 2 to 3 micron droplet size is being pushed with the piston through very, very small channels, and generating pressures of upwards of 30,000 to 40,000 psi, and then the resulting emulsion that comes out has a very small particle size. You can actually collect that emulsion and then push it through it again at a higher pressure or the same pressure, and you'll get a narrower and narrower particle size distribution in your final emulsion, and the emulsion will be more stable every time you pass it through. But usually, efficiency wise, you want to do it in one pass so you don't have to spend more time passing it again and again, because each pass could take a few hours to do, depending on the batch size. So right now, let's discuss the nanoemulsion droplet size or micelle size versus bioavailability. How does it help us to decrease the particle size in terms of the absorption in the body? So this, until 2018, phytocannabinoids haven't been, people haven't been able to do much in vivo studies, in vitro studies on them, because CBD, for example, has, until 2018 was considered a Schedule 1 controlled substance. So there hasn't been a lot of research on phytocannabinoids in the body. Since 2018, there have been some very few studies out there, but the data is very little, and not very, like 6 people, 10 people got together and did the pharmacokinetic study. So here I will constantly, because all the lipids behave similarly in the body, I will, I found a really good study that was done with the anacetrapib plasma concentration versus time, and this is in dogs in vivo. So this graph illustrates, perfectly illustrates how lipids behave in the body, even though it's dogs, but it behaves similarly in the human body as well. So here you have micron size, amorphous API. So it's basically dogs 8 micron powder. Then, the scientists, they encapsulated this drug in 1% vitamin E TPGS emulsifier and got a 347 nanometer, particle size. And then they emulsified in 2% TPGS, got 190 nanometers, then 3.5% and 10% TPGS, and got smaller and smaller in terms of particle size. And then looking at how these nanoemulsions or microemulsions and nanoemulsions behave in the body, you see that the 8 micron powder, it has almost no absorption. It's very little. As a matter of fact, it was measured to be 0.1%. The rest is just excreted. When you go into a microemulsion, so from macroemulsion to microemulsion size, you see a drastic increase in absorption. However, between 347 and 190 nanometers, there is statistically no difference in absorption. So, whether you're 200 nanometers or 400 nanometers, there's almost no difference there. Going further, decreasing down to nanoemulsion, below 100 nanometers, you are in nanoemulsion sphere. You are much higher than the microemulsion again, but again, between 3.5 and 10, so between 50 nanometers and 100 nanometers, there's statistically no difference here. So it's not a linear increase in absorption. It's like a stepwise process. I would equate this to, say, let's draw a parallel. We have an open door, and there are lots of little children running through the door. In the same amount of time, there are more little children running through the door. You can fit more little children than adults, right? Especially large adults. So the nanoparticles here are little children, and these sizes, the liquid crystal size or microemulsions are big adults. They can't fit as many. You can't fit as many of them through the door than small children. The micron size ones are basically like elephants, and so you could only hope to cut up an elephant in small little pieces, and only able to pass maybe 1/10 of what you've ingested, or actually 1/20th, or even less than that. So this is basically the basic process of absorption of lipids through the body. So now let's talk about the phytocannabinoid nanoemulsion pros and cons in foods and beverages. So up on the right side, you see the nanoemulsion with the less than 100 nanometer micelle size. It's transparent because light just goes through it. It does not get absorbed. Microemulsions are usually milky because the micelle size is large and the light gets absorbed, it gets reflected, and so on. So it doesn't go through. So you can't really read through them. Then another pro is obviously very high bioavailability, like we saw, it just goes through very easily. Then the superb stability. However, the cons of it is that the smaller the droplet size, the smaller is the lipid payload in the micelles. So the concentration of your active that you are trying to deliver is less and less and less, the smaller the particle size. Also, if your lipid is bitter, if you put it, if you nano emulsify it and put it in your mouth, it'll be even more bitter. Higher bioavailability, it gets to the bitter receptors much faster, and just bigger surface area. Smaller the particle size, the larger is the surface area, and that's why the bitterness just comes through a lot more. And more importantly, liver toxicity risk. So if your lipid active has even a little bit of liver toxicity, then if you nano emulsify it and take it at a large dose, at a dose where microemulsion would be safe, nanoemulsion might not be safe anymore, because now you deliver more of it in the liver and it might cause liver toxicity. So this is why with nanoemulsions, you have to be very careful. So emulsions with less than 100 nanometer droplet size. This is exactly why in UK and EU, the food regulatory agencies, they're not allowing nanoemulsions or nanoparticles to be introduced into their foods. They're very careful about that. You have to go through multi-million-dollar clinical studies to prove that you're safe and so on. So it's very difficult there to pass something like that through the regulatory agencies. And lastly, I wanted to talk about nanoemulsion, emulsions in pharma, food, and beverages. So they're used in foods and beverages everywhere, from frozen foods to ice creams and chocolate shakes, and some companies put vitamin C in liposomes so that they can deliver more of it systemically. Like Dr. Aust talked about before, Pfizer and Moderna vaccines contain mRNA encapsulated in a targeted liposomal nanoemulsion. So these are everywhere. Mile High Labs particularly creates very optimized 250 nanometer particle size phytocannabinoid liquid nanoemulsions that contain 20%, so 200 milligrams per milliliter of emulsion of phytocannabinoids like CBD, CBG, CBN. And so these are used in products like non-alcoholic beers and non-alcoholic drinks, teas, coffees, seltzers, soda drinks, and juices. This system can be used to encapsulate anything from adaptogens to like ashwagandha and other, mushrooms and stuff that have low bioavailability in the body. With this, this presentation is over, and I would like to open the floor for questions. Thank you very much for your attention today.